Hello, my name is Thomas Walsh from the Spanish National Cancer Research Center in Madrid, and today I'm going to talk about the APPRIS system for improving selection of principal isoforms, a project on which I've been working alongside Fernando Pozo Ocampo, Jose Manuel Rodriguez, and Michael Tress. Evidence suggests that a single main splice isoform reflects the biological reality of most protein-coding genes. APPRIS selects a single representative protein isoform for each coding gene based on cross-species conservation and the preservation of protein structural and functional features. Improvements to the annotation system mean that APPRIS core methods are able to predict a principal isoform for more than 75% of human genes, and the new Trifid algorithm produces a score for the likely biological relevance of both principal and alternative isoforms. Here we have a workflow diagram of the process of calculating an APPRIS score for the transcripts of a gene from the four component methods. Matador 3D scores each variant on its alignment to PDB protein structures. Firestar predicts functionally important amino acid residues. Corsair uses BLAST to score variants on their conservation over evolutionary time. And SPADE uses PFAM-scan to identify the set of functional domains in each variant. APPRIS assigns a score that is a weighted combination of the individual method scores, and on the basis of this, it selects principal isoform candidates. If there is one principal candidate, it is given an isoform flag of principal 1, the most reliable category of principal isoform. Otherwise, remaining principal candidates pass through a series of filters. Two sets of filters are currently used. The default filters are described in the APPRIS 2017 paper by Rodriguez and colleagues. The second set of filters, shown here, make use of Trifid, a machine learning classifier of isoform functionality, as well as Proteo, an APPRIS method that scores variants on their level of proteomics evidence. After each filter is applied, if one candidate remains, it is annotated as the principal isoform. So for example, if a variant has a Trifid score better than any other candidate by a sufficiently wide margin that it could be considered a dominant Trifid score, its isoform flag is principal 2. Otherwise, if a single variant has a Proteo score better than other candidates, it is marked as principal 3, and so on until a principal isoform is selected. For more on the Trifid selection process, please visit the APPRIS website via the link at the bottom right. To find out more about Trifid itself, check out its GitLab project via the link at the bottom left, or visit the poster Trifid Determining Functional Isoforms, also at ISMB 2020. The APPRIS database currently houses annotations for 10 ensemble species shown here. Among these, principal isoforms are also available for RefSeq reference sets in 6 species, including human and mouse. New species can be added on request. Here we see a breakdown of APPRIS principal isoform flags for the human genome in GenCode version 34. At right, we can see the breakdown for APPRIS version 31, which includes recent updates to APPRIS core methods, as well as the implementation of the Trifid transcript selection process. At left, we can see the breakdown for APPRIS version 29, the last version of APPRIS before these updates were implemented. Both have about 5,000 genes with a single variant, which are automatically selected as the principal isoform. Due to changes to the core methods, the new version of APPRIS predicts about 1,500 more principal 1 transcripts. With the Trifid variant selection filters, highly reliable principal 2 or principal 3 isoforms are selected for a further 2,200 genes. This slide shows a comparison between APPRIS principal isoforms, main select transcripts, and variants having a unique consensus coding sequence, or CCDS. Links to the main and CCDS projects are included at the end of this talk. In this graph, we compare each annotation set against the proteomics supported transcript for a common set of genes, all of which were annotated by APPRIS as principal 1. The technique of choosing the longest isoform as representative is also included as a baseline method. Orange bars represent percent agreement with proteomics data, while blue bars show levels of disagreement. As you can see, levels of agreement are very high between APPRIS principal isoforms, main select transcripts, and transcripts having unique CCDS. In conclusion, APPRIS principal isoforms are broadly applicable and are potentially useful in diverse areas of scientific research. Identifying the most functionally relevant or the most representative isoform is an essential first step in any genomic analysis. Following recent updates, APPRIS predicts a principal 1 transcript for over 75% of human genes in GenCode version 34 and identifies a highly reliable principal 2 or principal 3 isoform in a further 11% of genes using Trifid or Proteo respectively. In the human genome, APPRIS principal isoforms have high concordance with proteomics data, attaining a level of agreement comparable with main select and transcripts with a unique CCDS, while choosing a principal isoform for all protein coding genes. We would like to thank Daniel Cerdan Velez, Tomás Di Domenico, Laura Martinez Gomez, and all in the Cineo Bioinformatics unit. We'd also like to acknowledge the funding of the US National Institutes of Health.